Yeah, I've had my coffee. I hope you've had yours. Let's start. I'm sure you know people that are old money. Well, I guess I'm old coronavirus. It's all the same prejudices, none of the same prestige. But yeah, I'm the taller of the two people here. And this is back in 2004 when we were growing up. Tons of coronavirus, and you can see it's the fainter of the two bands here. That's a purification. And here's some of the little particles from a cryo-EM image. Now, before there was the most recent coronavirus, coronaviruses were a thing, but they were more an agricultural interest. So you've got these gamma coronaviruses. This is one of the big groups, and they infect all kinds of birds, but also cetaceans, so whales and dolphins, which is very weird. And you could say, what do these two things have in common other than seagulls? And I would say, I have no idea, but the thing here is that these are very distant branches and we don't have any of the intermediates in between. So there's some kind of wonderful story here that may or may not be lost to history. But coronaviruses definitely jump around. Now we've got more recent cases of jumping. So you've got viruses that are in pigs that are very similar to viruses in this adorable little bat, Scotophilus, up in the corner. And you've got another kind of virus in pigs that is a thing called a delta coronavirus, and it's related to the night heron coronavirus and a bunch of coronaviruses in less photogenic birds, but that's why we picked this one out. And we've even got closer relatives. So one of the human coronaviruses called OC43 is pretty close to the bovine coronavirus. They're actually considered the same species of coronavirus by the ICTV. And right there, kind of nestled in among these things, are some other viruses from the Hipposideros bats. And again, yeah, adorable little round leaf bats. But it seems as though these are viruses that are very good at moving from species to species, but they tend to make smaller steps. So we're jumping between chordate animals. We're, for the most part, jumping between different kinds of mammals. But I'm starting you off in the place that you know, and then we're going to go to the weird places because, yeah, we are all swimming around in an ocean of coronaviruses all the time. And thankfully, mostly we don't have to worry about them and we don't know about it, but they're everywhere. Okay, the latest viruses. Look at this cast of adorable characters. The little bamboo bat in the bottom corner just makes me happy every time I see it. These are some of the dangerous human coronaviruses, SARS-1, SARS-2, and Middle East Respiratory Syndrome coronavirus. And yeah, the viruses are very similar. These are all beta coronaviruses, and most of their relatives do seem to be in bats. And you could say, why bats? What have bats got against us? Bats are just really common in the world. About a quarter of all mammal species are bats from the latest species count. And so, yeah, it just stands to reason. There's so many of them. Yeah, it's only a few bats that are out to get us. And I would also point out that these are teeny tiny bats. Look, that's a fingerprint on the side there. And this is somebody's fingertips. So nobody is eating these bats. They're about the size of a chicken nugget. And I doubt they taste anywhere near as good. All right, but a virus is not its host, a virus is not its disease, a virus is its genes. And not all genes are created equal. Yeah. So if you want to know which genes are important, which, yeah, I'm sure you do, and I'm sure everybody does, a good way to figure this out is to just let the virus tell you. Compare a couple of viruses, and you can make kind of a heat map. And the genes that change less are generally the ones doing more important and general tasks. The ones that change more tend to be host-specific and tend to be adaptations to overcome the immune system, some particular quirk in your neighborhood bat or pangolin. And so looking at this, you can see there's a real cluster. Here we're comparing SARS to SARS-2, and here we're comparing SARS to Middle East Respiratory Syndrome. This is the big sector where you have most of the conserved genes. These are the enzymes that actually replicate the virus, and they're actually translated via a frame shift. That's why they're a little bit out of joint right here. But you can see some other areas, some other little hot spots at the beginning and some other little hot spots down at the end. And these are all interesting things. So let's look at a little bit of their machinery, and then I'm going to show you some new viruses that kind of fill out some of the early branches on the coronavirus tree and tell us maybe a little bit about where these things are coming from and what they may have in store for us in the future. So here are lots of, this is just the first chunk of the genome. So that first chunk before the frame shift. And what I've done is colored domains by conservation. So they're colored if they are encoding what appears within statistical parameters to be the same gene. And so you can see, what do these things have in common at the beginning of the polyprotein? It's a great big protein that's cut into little pieces. Not a whole lot. So one of those regions of conservation up in the beginning part of the genome is going to be NSP3 and 4 and 6. And they're important, but if you look between them, there's not a lot that you find in every single one, except that they all have these little black bars, which are transmembranes. And so that seems to be the defining feature. And you seem to be able to chop and change any of the other parts out and still get some version of a working coronavirus. Or in this case, we're broadening out to the nidoviruses, which is the bigger group, the order that holds coronaviruses. But look over here, and you see one of the universally conserved markers. So this is the main protease. This is one of our keystones. If we are identifying new species and trying to put them on the tree, that's going to be one of the 5 regions that we look at, because it's always there and you can always use it, and so it makes a real nice tree. All right, but what's a coronavirus really look like? So this is from a paper by Helena Meyer, and when the virus is outside the cell, sure, it looks like that little spiky ball, but when it's inside the cell, look how wonderful it is. It makes an organelle. It's a series of paired membranes and little blind chambers and open chambers. And these things, this is a electron tomogram, are where the virus actually makes more RNA. So it's going to manufacture things here. This is basically the virus factory. And those proteins that we just pointed out, NSP3 and 4 and 6, even though they have very little in common, those are the ones that seem to make virus factories in every coronavirus where it's been tested, and every nidovirus where it's been tested. And just recently, we had a wonderful new paper from Monza Barcena and Eric Schneider's group, and they showed that these double membrane vesicles actually have this wonderful pore on the top. This came out in Science maybe a month and a half ago. And the pore can let things out of the double membrane vesicle and let things in. Yeah, just like you would think. Now, they figured out that the pore had at least one of these in it. So they put a little extension up here and on the end of a protein called NSP3, and it appeared right here at the tip of the crown. So we know that the prongs are something like these 2 domains, maybe 3 domains at the most, just based on the amount of structure that's there. And we had actually, there was some old data that we had got together where we actually expressed these genes and showed that they really like to form pairs. Pretty much everything that's in this section will interact with RNA, which you'd expect, RNA coming out of a pore or possibly something going in. And they will also pair up, oligomerize in pairs. So we've probably got pairs that are making up each little tine of the crown here. And we think NSP4 is definitely in there because NSP4 is responsible for sticking the 2 membranes together. We figured that out from a couple coronaviruses. NSP6 looks like it ought to be part of the group. You can express it with these other things. It doesn't seem to much care about the other things. We are not sure where it's involved or indeed how deeply it's involved. And I'll show you there are actually some viruses that are lacking NSP6, but are still coronaviruses. So from digging through some of these new viruses, we were able to find a couple of new domains. But here, we'll show you the old domains. These are the ones that we look at in order to categorize these. You've got the NERAN is this little pink bit on the back. This is a nucleotidal transferase that's attached to the polymerase. You got the polymerase itself, which is kind of on the back side of this thing. And then the polymerase has a couple of factors. They sort of look like golf clubs. Here's the head up here, and there is the shaft of the golf club. And they form an exit tunnel where the newly synthesized RNA can come out as double-stranded RNA attached to its template. In addition to that, so we've figured out now that with all these new viruses, they seem to have proteins that you can't directly link to NSP8. They don't have enough conserved amino acids, but they're in the right part of the genome, and other parts of the genome are statistically similar. And so we can't call these domains per se because you can't justify them as that, but we call them landmarks. So there's a thing called the nidovirus helical landmark, and you find it in the same spot as NSP8 in every nidovirus that's out there so far. The other thing that we find is that you have the nidovirus metal binding landmark. And so it's going to be, it's going to look like NSP10 of SARS coronavirus. It's going to have 2 zinc metal binding sites in there and a couple of helices. And right up at the beginning, in between those, is going to have 2 or 3 beta strands. That seems to be a very common thing. And this is a cofactor. It activates both of the methyltransferases, and mutations in this seem to affect the function of the protease. So yeah, it gets around, yeah. All right, here's the second part of the genome, just to show you all the ones that we used to know about before the new guys come in. And you can see the second part of the genome, this is the part where there's the high conservation. It's also more conserved across the entire group. And so the coronaviruses are up here in this little bracket, but you've got basically the same organization down here in all these other nidovirus groups, some of which are found in flatworms, for example, some of which are found in insects, some of which are found in snails like this guy, and some of which are found in shrimp in the ocean. So, all right, and yeah, those are the other domains. So the other big domain that we look for is going to be the helicase, and this is a structure of the MERS coronavirus helicase right over here. It's got the usual helicase domains down here, and interesting middle domain that you find only in coronaviruses where there is evidence of a capping system. When the capping system is gone, the middle domain is also gone. For a long time, we thought it might have something to do with that, but cannot show it yet. Anytime you make a mutation in there, the virus is just dead. So yeah, it's been tricky. And at the beginning, they have a 3 zinc metal ion binding cluster right at the top of that helicase. In addition to this, what we think of as a coronavirus looks something like this. So it's got those spike proteins on the outside, it's got a matrix protein or membrane protein in the middle, and it's got a nucleoprotein on the inside, and it's got a little E protein that mostly gets left behind in the cell, but you get little traces of it in there. So I've drawn that one in horrible neon yellow here. And what we figured out from cryo-EM work is that these proteins, particularly the M protein, is determining the spacing of all the other proteins. So the other proteins slot into the M grid, and they can slot in at the nearest spot. This is the nearest that's allowed without a steric clash, basically. Or they can slot in at the next spot along, or a little bit later at the next spot along. And so you get variable structures for every single coronavirus, which is why you can't average them. You can only look at little parts and average those. And the M protein's job is appears to be to curve the membrane. So if you get all your M proteins together, each one imparts about a 6-degree curve on the membrane, and they have 2 different shapes. They have a compact shape, which does not curve the membrane, and a long shape. They sort of transform shape, and that one is curves the membrane and makes it very rigid, which helps the virus to escape, we think. All right. But that's just coronaviruses. If you look at some of the other nidoviruses, mostly they do not look like coronaviruses. So you've got your coronaviridae, and they have that spike, that M protein, that N, and that E protein, which is great. But look at some of these other crazy corona-like viruses, and they will have alpha virus-like proteins, or they will have unique proteins that nobody's ever seen before, or yeah, they'll just have some mixture of these things. The structural apparatus across the nidoviruses seems to be totally variable. They can chop and change. But we're, I think, able to figure out what probably the ancestral condition, the original one was. So let's go look. All right, so those are the coronaviruses we know. These are the coronaviruses that you're meeting probably for the first time today. So one of these is in a lovely Chinese frog right over there, Microhyla. Then we've got the axolotl, which has a giant coronavirus that's related to these, and the Chinook salmon. All 3 of these are very early roots on the coronavirus tree. These guys actually branch out before you get to things like the chicken coronavirus or anything like a bat coronavirus. So whereas we may get viruses from bats and other animals, those animals eventually, if you go back far enough in time, seem to have got them from the ocean, hence swimming in the sea of coronaviruses. So here's a little look at one of these things. And interesting thing about this particular virus is it's really prevalent in the very young tadpole stage and declines through metamorphosis. By the time they get to early maturity, it's almost gone. And when some colleagues went back and looked at maturity, they were unable to find any trace of the virus. So it's a baby frog virus. This is kind of like one of those colds that your kid is going to get at school if they go in person at school, and bring home and get over, and then get again in a year or two. And yeah, frogs get these too. The virus itself is fairly coronavirus-like in the 3' half, I would say, of the genome. It has all the expected enzymes and has the usual coronavirus-like set of structural proteins. But in the front part of the genome, the ones that make up that giant pore, those are very, very different proteins. They look like they're from outer space. They are really neat, honestly. But the other thing that we can tell just from pulling these sequences out of metagenomic databases is that there are these jumps. And so everywhere that I've got an arrow is a place where we're expecting it. But you can see some of them didn't jump very much. Yeah. What are these? Well, these are where we expect there to be more RNA. So the way the virus works, it has a genome. It makes it into a series of negative strands, subgenomic length templates, which have the same beginning and the same end up to a certain point, and then leave out one or more genes toward the 5' end. And you have the full genome, so you make a full set of these things, as you can see over here on the right side. And because of this, if you're just reading these out as sequence read depth, what you would expect is there will be the most of the things down here at the 3' end and the things right at the beginning, something like this. So here we've mocked it up. And this is a wonderful paper. It appeared in Cell, I think back, March or something like that. And this shows that for SARS coronavirus 2, it's got the same little stair steps, and everywhere that there's a jump, that is a subgenomic RNA. So that's what it's telling us. And this is a feature that unites nidoviruses together. They make these subgenomic RNAs in this particular way for the most part. Let's go to a deeper branch on the tree. So that's an example of the ones in the salmon and axolotl and frog. Here's a weird branch. So this is in a giant mollusk, the gastropod Aplysia californica. And this wonderful creature, they get about as big as a bread roll, and you can hold them in your hand. These are a neurological model. They are, I just think very charismatic little animals. And every single one of them in captivity appears to be infected with the same virus, which we found and named Aplysia abyssovirus, because it comes from both deep sequencing and like an ocean animal. It seemed like a cool name, yeah. And you can see the jump there with the subgenomic RNA right over there. All right. And so even though it only has one structural protein, it seems to, it commits to it and it makes a messenger RNA for it. So it's just like a simplified version, which is also much larger than your standard coronavirus. But since this is a sequence that we're basically pulling off the internet and other people's data, we wanted to see, is this thing even real? And so we made some of the parts of it. And so we made the region around that main protease, that was the blue chunk before. And sure enough, it's able to cut and free itself from a larger piece, which is shown up here. And when you mutate out one of the catalytic cystains, then it no longer is able to do that. And so, that's exactly what you would expect. We also found that instead of a normal frame shift signal, this particular virus has a stop suppression or a stop skip mechanism, in that it has an in-frame stop codon, and it has this wonderful secondary structure up here that is able to remove the effects of that stop codon. So it's able to get the virus to be translated in the second open reading frame and actually make that polymerase part, which is unusual. Regular coronaviruses don't do it that way, but they have something similar. And this is an even weirder branch. This is the latest one from this particular group. It's from Yuri Temura. And the thing that it is missing is it doesn't have an equivalent of NSP6. It should be right over here after this little blue box, and it is just not there. So the protein that we can't figure out what it's doing, but it seems to be part of the pore, is apparently sometimes not part of the pore. Or at least there's nothing that has a transmembrane predicted in that region. It's nice and tightly packed with all the other things that we would expect, but and none of those. We do find things like the nidovirus helical landmark, the NIHEL, and the nidovirus methyl binding landmark. Those are definitely there. And you can see it's got two of what appear to be alpha virus-like spikes. So this is probably going to be an icosahedral coronavirus, which is something that I thought I would never see, but there's one. Okay, But what if we take an even broader view? It's not just coronaviruses, and they don't exist in a vacuum. There is another group, and I think we're going to name this the Proto-Neato Viralis. They're going to be like nidoviruses. That's who they're closest to. They seem to be so far just in arthropods, a variety of insects. This is a little tick over here. Sorry, a little, little mite over here, and lots of spiders. Yeah, if you like spiders, this is the group for you, for sure. And so the color scheme's a little different, but some of the genetic parts are the same. So you can see we've still got our polymerase here, which is fine, like a coronavirus. After the polymerase, we've still got a helicase, like normal. But the helicase doesn't have that metal binding domain, completely gone. You cannot find 4 cysteine or histidine residues anywhere near each other that even could make a metal binding domain. So it's possible to exist without. And it's missing that middle domain that's associated with having the other capping enzymes that would normally come after. And then the polyprotein stops. So it's, polymerase helicase done, instead of a normal little train of 5 or 6 enzymes there. In place of the usual apparatus, it's got one multiply transmembrane protein. Look at all those little black stripes on there, like a little tiny tiger. And so this one, if it is making a pore, this is probably the pore-making protein. And it does have that main protease here, so that's still the blue square. And so it's got 3 out of the 5 nidovirus-specific things, and it does have those 2 other nidovirus landmarks. It has the metal binding landmark, which is in these 2 groups at the top. It's the sort of light blue. And it has the helical landmark, which is in every single one of them, which looks like NSP8, and it's probably the exit tunnel for the polymerase. So this is a really primitive branch on the way to coronaviruses, or with a common ancestor to coronaviruses and nidoviruses. And very different. Now also look at the structural apparatus. So whereas in nidoviruses, we get this wide range of structural apparatus, in this other order, which each of these little sections is going to be its own family. So these are the first 5 families in the Proto-Neato Viralis. There's only one type of structural apparatus. And it's a thing that we call, well, that we're planning to call the MES protein, because it's going to have the same function as the M protein of coronavirus, the E protein of coronavirus, and the S protein of coronavirus. And we come to that logical inference because there are no other candidates for these proteins. And it looks like a polyprotein. It's got 2 big regions that could be sort of spike-like. And the final region here is actually pretty well conserved. And in some other viruses where we find this, we do see that this is actually related to the coronavirus spike. So the actual coronavirus set appears to have taken this last gene, deleted all the front, or just split it up into 3 different proteins, which is kind of neat. And it's got a nucleoprotein which overlaps at the very 5' end of that gene. That's the little orange hashed bar here.
There's 2 minutes left, Ben.
Perfect. Yeah, I only need 1. Yeah. All right. Let's see. So MESs, interesting. Here they are. We also find them in some coronaviruses. They're weird coronaviruses in shrimp and in another kind of aquatic. It's like the little tiny aquatic bug. I think it's an amphipod. But yeah, these do exist, and we also find it in a lot of insect Misoni viruses. So since this is the one thing that we find all over the tree, even in viruses that have very little else in common, the idea is that this may actually be the ancestral set of genes. And so you'd have a case where you'd have this sort of multifunctional do-it-all protein that would be the original progenitor structural protein, and back when you only needed 1 in the good old days. And then maybe you had a nucleoprotein, and we see that actually does overlap. So those must have come in, they must have a long history together because they overlap almost completely in all of these cases. And then you get a sort of a coronavirus-shaped little tubular sausage of a virus, which then goes on to split up, diversify, and adapt to its hosts. And that's how we got coronaviruses. So thanks very much. Here's a pretty glass structure from, I think, Luke Jerram of coronavirus, and those are some of the collaborators. Happy to take any questions if there are any.
Thank you, Ben. And I'll just remind participants, if you have questions, please raise your hands, and we will allow you to talk. And I think Ray Silver may have a burning question. Ray, you can talk. Please unmute yourself. Ray? Maybe not. If not, Rodney Rothstein. Rodney, go ahead.
Yeah, very nice. Thank you very much for that introduction to all these viruses. I wanted to ask you about a specific protein that you didn't mention, and that's NSP14, which is the nuclease and the methyltransferase. And when did that appear, and does it really help make the virus less mutable, et cetera, as people think?
Yeah, great question. This virus, in addition to being a this enzyme, in addition to being a proofreading enzyme, Mark Dennison's group showed that it actually is important for the virus to make that nested set of RNAs. That said, there are quite a few viruses that seem to be lacking NSP14, this exonuclease and N7 methyltransferase combined, or that seem to be lacking one or the other domain. You'll sometimes find just the exonuclease and it'll be missing the capping apparatus. So the capping apparatus seems to have come in as a block. That's the second half of NSP14 and NSP16 seem to have arrived together. NSP14 seems to be like NSP15, the front end of NSP14, in that it appears in some lineages. It's there in most, but not all nidoviruses. It's missing from some of the invertebrate ones, and it is not there in any of the Proto-Neato viruses. So this is after the proto and the regular nidovirus split. Yeah, we think. Thank you. Good question.
Ben, if I could ask you a question, given the prevalence of the coronaviruses throughout biology, to what extent does recombination between closely related coronaviruses drive the emergence of new strains?
Every coronavirus genome is actually recombinant. Yeah, that's the thing. Yeah.
Yeah.
So people are saying, oh my gosh, it's recombined. You say, yeah, of course it recombined. If you put two coronaviruses into the same cell, they'll do a thing called leader switching, where they will actually, one of them will decide it's dominant, the other will be submissive, and the dominant one will put its leader on both of those things. And it is wickedly efficient. There's just no room for error. And so we think that every strand, even the genome, is discontinuously made. And they have this built-in jumping mechanism where they can stop with one, jump to another, and they do that to make their subgenomics. So I'm surprised it isn't more common, but I think for that to happen, you have to get a host that has two different coronaviruses in there. And that's probably all it takes. It's probably a near certainty when that happens. So yeah, we think this is probably very prominent. And thank goodness we don't know about more of these yet, but I am certain there are more out there.
Okay, and if I could ask one more question. At the beginning, you showed a beautiful EM picture of what you termed a virus factory. And I was curious at what point following infection, initial infection, do those organelles form, and what exactly is going on inside of them?
Yeah. The first time I've seen anyone detect these is around 4 hours after infection. And you can actually detect new virus RNA before you can detect the organelles that are supposed to make it. The double membrane vesicles seem to form first, and then within an hour you get what they call convoluted membrane in between and all the other little bells and whistles. But the neat thing about this is, if you look at different strains of virus that are temperature sensitive or different pathogenicity or just different strains of the same thing, quite often there'll be one that makes far fewer or far more of these double membraned organelles than the other ones, and they still seem to replicate their RNA just fine. So it looks like some kind of anti-host adaptation. It's common to all the nidoviruses. They do make RNA on the inside, and they do have all their replicase proteins associated with it, except I think NSP1 does not get associated with that. Yeah, why exactly they go to all this trouble and spend roughly a third of their genome encoding the machinery to make this lovely structure is still, I think, anybody's guess.